So we are discussing about flagella in bacteria. Let me adjust my little whiteboard. Okay, I think it's good now. So flagella, the function of flagella in bacteria and eukaryote is same. They are responsible for the motility of the cell, but the physiology for motility is different in bacteria and eukaryote. In bacteria, flagellum rotates and provide motion. So in bacteria, the motion of flagellum is rotatory. In bacteria, motion which is provided by flagellum, or this motion is rotatory motion. And in case of eukaryote, it's lashing movement. Let me draw a diagram. Let's suppose this is a bacteria. It has DNA, and this is basal body for anchorage of flagellum, and this is a helical shape of flagella. But in case of eukaryotic unicellular organism, let's suppose there are some flagellates, you can say Chlamydomonas. In Chlamydomonas, there is a single polar flagellum. They will also have a basal body for the anchorage, and they have long flagella. Now, in case of bacteria, this flagella rotates and provide motion. It will rotate and provide rotational motion. But in case of eukaryotic flagella, they keep on lashing their flagellum right and left and provide motion. So, but in case of bacteria, the flagella rotates in its position, and this rotation provides energy, provides a push, provides motion to the bacteria, and bacteria can move. But in case of eukaryote, the flagella is not rotating, it is standing in its basal body, anchored. So they will keep on lashing their flagella, and due to this lashing they can move. But in case of bacteria, the flagella rotates, and this is called rotationary motion. So this is the difference of physiology of flagellum. So how they move, but flagella, the function of the purpose of flagella in bacteria and eukaryote is same. They are used for motion. So let's talk about the general structure of a bacteria. Let's suppose this is a bacterial cell. I'm going to draw a general structure of a bacterial cell, so that in our further explanation you could be able to understand. And these are basal bodies, and here is your helical flagella or singular flagellum. Listen carefully. This is genetic material of a bacteria. This is called plasma membrane or cell membrane. Okay, let me write it as a plasma membrane. In plasma membrane, everything collectively known as protoplasm, and in case of bacteria, plasma membrane and everything within plasma membrane is collectively called as protoplast. I think you are well aware of these terms in your previous classes. So outside the plasma membrane, if it is a gram-negative bacteria, listen carefully, if it is a gram-negative bacteria, outside the plasma membrane there will be a semi-rigid structure that is called cell wall. That's called cell wall. So this is cell wall of bacteria. Now bacteria is covered with a semi-rigid covering that is called cell wall. So this is gonna be the cell wall of bacteria, which is composed of peptidoglycan. If you see the composition of cell wall of bacteria, that is composed of peptidoglycan. We will discuss this component in great detail actually. So for just now, you can say this is a peptidoglycan layer which acts as a cell wall. So in case of gram-negative bacteria, there is a further covering present outside the cell wall. Okay, this is very close to cell wall. There is a further covering present outside the cell wall. This is called outer membrane. So this is called outer membrane.